Just want to let you know that the first one was a brand new species of octopus that was just filmed last summer. This one has been filmed a while back, but it's a new species found in the vents. And you'll notice that it's webbed-footed. It's one of the only webbed-footed octopi ever found. In fact, we find octopi at hydrothermal vents that we don't see anywhere else in the ocean. So consequently, we have a whole new octopus group. This is when I normally played a Bach or some other kind of ballet music because it's sitting on top of a dead black smoke or spire where it's dancing, basically. I happen to love octopi for a lot of reasons. They're the largest brained invertebrate. Unfortunately, they only live to be about 3 or 4 years old. I think if they lived longer, they would be the most intelligent being in the ocean, probably. Interestingly enough, the genome on these animals, on an octopi, has been sequenced. And it turns out, for example, the eye, and we've talked a little bit about the eye and these conserved genes, almost 3/4 of the genes in the eye and the octopus are virtually identical to our eye genes. So we are very closely related, at least by eyes, to these octopi. So anyway, I'm going to try to finish the stuff on Extremophiles and by tomorrow actually get into origin of life stuff. And so we covered high temperature this morning, and I want to cover the low temperature end. But before I start, we had this discussion about the phylogenetic tree and whether or not there might be some kind of shadow biosphere. And so I actually had been thinking about this for a long time. I had published a paper on this in 1998 about alternative trees. This is essentially the tree of life as we know it, but could this core be a branch from an early tree that just became extinct? And so are we making a mistake in inferring that the earliest organisms that ever existed on Earth were, the organisms growing in extreme environments? On the other hand, there might have been lots of extinct type patterns, and perhaps there is a shadow life branch somewhere right now that we haven't detected. I'm not too keen on this, but I've placed it in there as a possibility. So if this kind of tree existed before this, it was very, very short-lived. And this would mean quite a bit of diversity would have had to happen in a very, very short time. That might be possible, but if it is possible, these guys would be quite different from what we know about these guys in terms of how they change genetically significantly. And so to get back to the low temperature, this is just looking at the range of low temperature microbes. There is growth at -15. It may eventually get colder than that. And they generally have their best growth temperatures under 10 degrees Celsius. And then there's groups that actually grow at 0 degrees, but at much higher temperature. These are cycrotolerant. The cycrophilic and cycrotolerant organisms are very, very important with regards to planetary protection on icy moons. And so there's a lot of concern about them and whether or not there are any of these kind of organisms that can actually contaminate a space vehicle and reach. This is a topic we can talk about because some of these actually, particularly these cycrotolerant organisms, form spores that are resistant and could actually potentially survive. So right now, the lowest temperature for growth is -12. Active metabolism has been found in ice so that microbes are still metabolizing down to -22 degrees Celsius. And there's some really beautiful work that's been done on that. What happens with sea ice as seawater becomes ice, the particulate material and salt start forming briny regions, and you end up with channels throughout the ice that are filled with brine and organic material, so it concentrates that, and that becomes a really hotspot for microbial activity in the ice, even though it may represent only 1 or 2% of the overall space in the ice. Evidence for survival at temperatures as low as -80. In fact, we preserve our organisms in liquid nitrogen at -80. So there's no question about them surviving very low temperature. Spores have been found in ice cores that are over 1 million years old. There's actually controversial, but there's spores that have been found at over 25 million years old. I'm not sure we want to believe that, but certainly there's pretty good evidence for around 1 million years. Spores have been found in very ancient amber also. So these organisms can survive some really extreme sets of conditions. One of the first of the very cold organisms also found in sea ice that grows at -20. It actually has its optimum growth temperature. That means it grows at its best at 4 degrees, and that's Ramonas. And that was isolated by Jim Staley, who was actually a member of our astrobiology program at University of Washington. Since then, Ramonas in Grammii, which now has essentially one of the lowest growth temperatures at -12, was isolated from permafrost. And I'm just showing these to show you some pictures of a group of cultured low temperature microbes from a variety of different authors. All of these organisms have an optimum growth temperature below 10 degrees. This is Coelia psychoerythra, named after Rita Caldwell, who was, I don't know if any of you have ever heard of Rita Caldwell. She was the head of NSF for years and is a marine microbiologist. And I got my PhD from the same person she got her PhD from, so I have a little bit of loyalty towards her. And the other thing that's kind of interesting in this picture, see these guys here, which are about 200 nanometers. These are viruses that attack these bacteria. So this is what a virus looks like. It has a head made of protein. The nucleic acid is in there, the tail. And we'll talk more about these. These are fascinating creatures. And it turns out that in ice, for example, when you concentrate bacteria, you also concentrate a lot of viruses. And there appears to be a very enhanced rate of horizontal gene transfer. That means genes being transferred to these organisms through viruses. When Jonathan mentioned that microbes don't have sex on his first that's wrong. They have lots of sex. They do it in 3 different ways. And if I have a chance, I'll show you those 3 different ways. And probably are the origin of our sex. So I know I hate to say that we're all just, came from microbes, but we certainly have. So if we look again at Acidophiles, the problems are preventing ice crystal formation. And so what happens, and when you can freeze a lot of organisms, if you have ice crystal formation inside the cell, inside the cytosol, it could actually break apart macromolecules. And so organisms that actually do this produce some kind of compatible solutes. That is, they'll produce an organic compound. They produce antifreeze, basically. And a lot of the animals that live under these conditions also produce antifreezes to prevent freezing. And then one of the main ways is the production of these sugars, these complex sugars, which surround the organism and protect them from that. To get protein activity and et cetera to work at those low temperatures, they actually have very, very different kinds of proteins that are set up to have much more flexibility at cold temperatures. And then the same thing with their membranes. Their membranes tend to have what we call more unsaturated fatty acids. That means there's more double bonds between the carbon than single bonds. And that seems to be a hallmark of those kind of organisms. Now, this is another interesting animal that was found as a result of the census of marine life. This is an Antarctic ice fish. It actually about -2 degrees Celsius is where this animal is found. It's the only known vertebrate without hemoglobin. It actually has no blood at all. And essentially the serum is just a transparent sort of saline fluid. Their metabolism relies on oxygen dissolved in the liquid blood and is absorbed directly through the skin from the water. And this works because there's a very high concentration of oxygen because it's very soluble in cold water. So this is a unique adaptation. It's the only vertebrate we've ever found without blood. So it doesn't transport oxygen to the cells in the normal way. As I said, anything is possible. So in summary on temperature, to date, the lowest temperature for growth, as I mentioned, is minus 12. And right now it's about 122, but I guarantee you within the next year it'll be higher than that. But I won't reveal that yet. Low temperature microbes do not have ancient lineages. There's nothing ancient about any of the organisms that grow at low temperature. I mentioned spore-forming psychrophilic bacteria are very much a concern in planetary protection issues. And how do we get rid of them? Where might they come from, etc. High temperature microbes have ancient lineages, and that's one of the reasons we study them. We've learned a lot about the origin of metabolism and the origin of eukaryotes from these organisms, and we'll get more into that. And then I also mentioned the fungi that grow at 60, but 50 degrees is about the highest that a multicellular organism can grow at, and that's a hydrothermal vent worm. So we move on to pH, and this is just a spectra from pH 0 to 14. You can see at pH 0 there are archaea, fungi, and even a eukaryotic algae that can grow at pH 0. So that's not seemed to be an issue. There are actually animals that grow at pH 2. They can handle low temperature. Even goldfish can handle, like carp, can handle pH of around 4 for a while anyway. But as you get into the, and of course everything grows in this range. As you get into the more basic environments, there are algae and bacteria that grow in these range. I'm going to be talking about the only marine environment ever found that is around pH 11 or so, fluids that's also above 90 degrees. It's the only one in the ocean that's both high temperature and high pH. And as you would expect, this is a combination of extreme conditions that's very, very difficult for organisms to grow in. So it's a very interesting group that grow under these kind of conditions. There are lakes called soda lakes that are very high on calcium carbonate, etc., that are also very high pH. On the low pH range, Thomas Brock was the first to isolate, and I mentioned Thomas Brock in the 70s, to isolate an organism called Sulfolobus, which grows at pH 1. This is an acidic mud pot in Yellowstone Park, and there's many of them around, including ones in Naples. These are organisms that actually use oxygen and take sulfur, either elemental sulfur or hydrogen sulfide, and oxidize it to sulfuric acid. And so they're growing in, several molar sulfuric acid. And so they have a mechanism to do that. And this is what Sulfolobus looks like. It actually is what we call pleomorphic. It takes on a lot of shapes. It doesn't have a rigid cell wall. It actually has a special kind of envelope that it grows in. The record holder for low pH is Picophilus, that grows essentially down to pH 0.06. And I don't know what pH 0, or even in this case, there's a report of this growing at pH -1. I don't know what that means. That's very acidic. It grows at moderately high temperature and was isolated as sulfutaric soils, which are a lot of them associated with geothermal regions. And this organism maintains pH membrane integrity at pH higher than 4. And this is what it looks like. I'm just showing this as a growth curve because I always find really remarkable, this was when this is the first time this particular organism was displayed, it can't even grow at pH 3. That's what's so interesting. I just never knew an organism that would be that way, and its optimum growth is under pH 1. And so it's totally adapted to these low pHs. It's just very unusual. Optical density means when, if we just put cells in a spectrophotometer, increase in numbers can be shown on the spectrophotometers, and this is growth over time in hours. So some of the unusual characteristics, in 2004, Picophilus had its full genome sequence. It's one of the smallest genomes ever, tiny, tiny genome, 1.55 megabase pair is very, very small. Take E. coli, which has about 3.6 megabase, for example. And then a very high ratio of genes encoding a proton motor force, and I'll describe that. This is basically how you can derive energy by living in a pH gradient. So if you're living at pH 0 and you actually can maintain pH of 4 or 5 in the cell, you could take advantage of that pH gradient and get energy from that. That's what a proton motor force is. High coding density, that means it doesn't have a lot of wasted genes. 91% of the genome coats protein. Even typically, most bacteria, we rarely see more than 40 or 50% of the genes transcribed at any one time. And of course, very acid stable membrane proteins, and I need to go into that. Something I'll talk about later. The first paper to ever show viruses for high temperature archaea was in 2003, and that was at Yellowstone. And since then, there's been several other studies. The most bizarre shapes of any viruses, you can't even imagine the shapes of the viruses. I'll show them later. I mean, even in my weirdest imagination, even if I took drugs, I could never have come up with some of the shapes of these viruses. And of course, I would never take drugs. So I have to just rely on my own normal self. So in acidophiles, the whole idea is making sure that internally your pH doesn't get lower than about pH 4 or 5. Otherwise, it just totally denatures things. So when you work with these organisms, and I've done that, there's two things you have to do. You either have to take them out of their environment and put them into something that they can grow in, or if you want to preserve them without growing them, then you have to put them in a neutral solution, pH 7, so they don't grow, but it doesn't affect their internal pH. Protein stability, protein in the wall of membrane are made to actually withstand a really low pH. And again, this proton motor force is where they derive a lot of their energy. And I thought I'd put this in here because we're going to come back to chemoosmosis. This has been a big, big thing about early organisms and the origin of life. And so if you get an ion gradient, let's say protons, it has the potential energy that can be used to power chemical reactions if they're forced through some kind of a channel. And essentially, photosynthesis and most of the heterotrophic systems go through this kind of reaction. And so it's basically protons moving through this and then through oxidative phosphorylation generating ATP. And essentially, that's a light-driven and chemical engine. If you have to have a pH gradient, you can actually have protons going in and out on a pH gradient and actually derive a free lunch. If we move on and go to growth at high pH, Halophiles grow at greater than pH 9. The record holder is a Neutrinobacterium that has an optimum of 10, grows at 11.5. And these grow in these various soda lakes. There's some in Egypt, we have some in California, but the sodium chloride can be different, as is the sulfate, but very, very high carbonate and bicarbonate in these, and so it results in very high pH. The properties of these organisms, many are uncultured. We don't know very much about them, but we know that they exist in these kind of environments. Some are well-known species of bacteria and archaea that have been adapted to this. Some can grow very, very fast. This is what's really interesting. They grow under these conditions. Doubling time of 10 minutes. Did anybody actually do that calculation that I mentioned? What did you come up with? It's more than the weight of the Earth. And so it's a good calculation that if something doesn't, if there's something not limiting a simple microbe growing at, let's say, 20-minute tubbling time, if you keep it going for 2 weeks, it will exceed the weight of the Earth. So clearly there are some controls, obviously, in this. But it points out that during, I mean, the whole reason that I wanted somebody to do that calculation was that it points out that I think during early Earth history, when new metabolisms were just starting out, and so, let's take advantage of this particular nutrient. At some point, the enrichments would have been extremely great before it would run out of some limiting nutrient. And we see that today. I saw that at Mount St. Helens. When Mount St. Helens blew up and all the mineral nutrients that came in on the tephra, ash material, I had microbial biofilms that were that thick. I mean, there was nothing to eat them. And in a matter of 2 months, there was just gigatons of microbial biomass. And then, of course, things came around to eat them. But I really got a first sense of what it must have been like during a time when there wasn't things to eat microbes that really must have taken off. So they can grow very, very fast. There's some that we call triple extremophiles that can grow at high temperature, relatively high temperature, high salt, and high pH. So the problems, again, is internal pH. How do you maintain a pH that's lower than 9.5 so as not to destroy your cell? Stabilizing proteins. Many of the alkalophiles also are dealing with high salt, and how do they deal with that? And I guess bioenergetics, essentially, all of them have adapted to this proton chemoosmosis system of deriving energy. That seems to be one of the first strategies when you're growing at these various high or low pHs, is how do we take advantage of this proton, of the protons in a pH gradient. This is Soda Lake. This is Mono Lake in California. It's a pH of about 10. The towers are calcium carbonate. I chose this particular black and white picture because it's quite artistic. It's better than some of the funky color ones that come out. It's about 8.5% various salts, not just sodium chloride. And there are brine shrimp and diverse groups of microbes in this lake. Why is this, I'm showing this, is because the paper, a paper came out in 2008, is high amounts of arsenic in Mono Lake. And Ron Omerlin's group actually found for the first time a group of photosynthetic organisms that don't make oxygen, but actually use arsenate as the source of electrons. So how this is a new electron donor for CO2 fixation. So again, cyanobacteria and green plants get their electrons from water. They split the water. And then a lot of anoxygenic photosynthetic bacteria use hydrogen, H2S, iron, nitrite, and now arsenate, using the Calvin-Benson cycle. So this was a new find in which you can now use arsenate as an electron donor for reducing CO2. And for those of you not familiar with this, I thought I would show all three kinds of CO2 fixing pathways. I mentioned in green plants and cyanobacteria, light, you split water. The hydrogen is then necessary for reducing CO2 to make what Jim Castings thought biologists never use, which is CH2O as a way to depict organic carbon, and they produce oxygen. In anoxygenic photosynthesis, it can use H2S or hydrogen or iron, and now arsenic. And that is used basically to reduce CO2 into that. And in case of hydrogen sulfide, you produce some kind of sulfur compound. And later on, I'll talk about when this kind of group of microorganisms arrived, because it came way before this group. And then chemosynthesis, again, is here using oxygen with hydrogen sulfide and deriving enough energy to fix CO2 from that without light. And besides the use of hydrogen sulfide, some of these microorganisms use chemolithotrophic or chemosynthesis organisms use hydrogen, methane gas, and a variety of metals like iron and manganese as sources of energy. So they use that with oxygen to reduce CO2. So this is essentially the CO2 reducing group. So these electrons are available for photosynthesis by microbes. This is the reaction of arsenic to arsenate. And this particular red pool here are where the arsenic photosynthetic microbes are, and they look like this. So this is, again, a first. But then I think this helps stimulate this work that I'm sure most of you have heard about. It's Felice Wolf-Simon. Ron Orman is also on it. It was published in December of 2010. And can arsenic substitute for phosphate nucleic acids? Keep in mind that one of the people here is also Paul Davies. And I mentioned Paul Davies, who's very interested in shadow life. And he was thought that an organism that can actually use arsenic instead of phosphate as the backbone to DNA and RNA would be such a shadow life organism. Well, as you know, NASA made a very big deal about this and released a press release days before it came out. I had about 30 emails from press all over the world saying, did we discover life on another planet? You know, are we starting to talk to intelligent life somewhere? I mean, it was so blown out of proportion. And then when it came out to be an arsenic thing, it was, I think, a little bit anticlimactic. But this particular work also has been scrutinized fairly seriously. And I'll point out my own issues with the arsenic paper. Right off the bat, they state that there are no prior reports of substitutions for any of the 6 major elements essential for life. And you heard that in the first lecture from Jonathan: carbon, hydrogen, oxygen, nitrogen, et cetera, the CROMPs, they call them. And this is wrong. There are 2 prominent examples. There are arsenolipids that have been known since 2004. So in some microorganisms, in the absence of or low phosphate, arsenic can form a relatively stable bond with the lipids. And there's been a couple of papers since. Other thing is selenocysteine. There is a amino acid. This is a sulfur amino acid with selenium on it. Selenium rather on it. That is very involved in certain key enzymes. So this is like one of the 22nd amino acids that is found in life. And we've known this for about 20 years now about selenocysteine. They also discussed the idea of an alien organism. The organism they work with is actually very common. And it's a late branching bacteria known to tolerate high concentrations of metals. It's kind of personal to me because I have actually named 4 species of that same group of organisms, the Halomonas, and I've studied them quite a bit because they're found also in the vents. And a couple of things: they tolerate really high concentrations of metals. That's something they do. It's also very difficult to achieve phosphate limitation in microbes. They just are very creative ways of storing phosphate. And in their work, there's actually some phosphate that was present in the medium, which is actually enough for, I think, a number of organisms to grow. Now, I want to point out, I think this was a great idea to run, but this is also a very good example of not being very critical about the results and not going into the literature and seeing what was already done in this. In terms of phosphate, I have tried for a long, long time to get get some kind of medium for microbes from the vents without phosphorus, because what I want to do is add the mineral apatite, which is sort of a P2O5 mineral that is in the solid state, to see if microbes can dissolve that. And so whenever I try to make up a growth medium without phosphate and I throw in apatite, I get the same growth as I get without throwing in apatite. They just simply really have a way of scavenging it. And I also point out the creativity. Cyanobacteria, for example, the photosynthetic organisms in the ocean, the dominant CO2 fixing organisms, have been discovered just last year that when phosphate is limiting, they actually incorporate sulfur into the lipids that are normally phospholipids. And so that was just also reported. So they saved the phosphate for DNA and RNA. Anyway, just last month, or last week rather, a paper came out in a journal called BioEssays by a group of people who actually do a lot of arsenic work. And so they have a complete literature of what is known about the arsenic in biology, in particular where arsenic can substitute for phosphate in living organisms. And again, it can in lipids. However, the authors point out that there is an extremely rapid rate of spontaneous hydrolysis of arsenate esters. That is, when arsenate is associated with acids like C double bond O, that would make the small molecules such as sugar arsenates very unstable. And arsenic DNA and RNA rapidly fall apart because the bond is really very unstable. So the authors conclude that considering this later consideration indicates that arsenic life seems very, very unlikely. Essentially, arsenic on DNA or RNA just falls apart. So I just want to point out, I'm pointing this out because, you know, as somebody really committed to astrobiology, I don't want this to set us back.
So has this paper ever been published as a paper? I've only seen it in Science Express, but never in Science.
Well, that's Science Express is regarded as a paper, as a legitimate publication. So it's reviewed and everything. It's just a more rapid online version of Science. So, but it's a legitimate paper. Within the next week, supposedly it was supposed to happen by the end of March, but it looks like it's going to be early on in this month. Science is going to publish all of the letters that came into Science regarding this paper. The last I heard, there were 12 substantial letters that came in, all negative. And so they've given the authors of the paper time to work with these 12 and try to respond to them. So it'll be interesting to see what happens there. I feel sorry for the authors in a lot of ways, because this is a tough thing to deal with. So dry environments, again, related to high salt stress and the availability of water. Most soils during summer months, for example, get very, very dry, and that's why many of the microorganisms in soils form spores to deal with the drying. They form spores of resistant cyst. The Antarctic dry valleys and high salt lake deserts, for example, are very, very well studied by the astrobiology community as sort of Mars analogs to see what can actually work there. I'm not going to cover everything, but I would like to say a little thing about desert Varnish, where we actually see black coloration on rocks, and it's a precipitation of iron manganese deposits. And microbes do that, even though there's just very, very little moisture. So there's a variety of rock-hosted microbes. I'll point out one of the great examples of a life form that grows under very dry conditions are lichens. How many people know what lichens are? They're algae and fungi, so they're a symbiosis. What you might not know is that the first person, anybody know who the first person was to identify lichens as a symbiosis, as something between two different organisms that were codependent? Who? Nope. Nope. I'll tell you. Beatrice Potter, who wrote all the little children's books, she was the first to actually come up with that. And so she had this great idea, and she's an English author who wrote, what's the name of the children's books that she wrote?
Peter Rabbit.
Peter Rabbit and a lot of those, Beatrice Potter. So, I mean, those of us in the English language world were raised around those books, so I knew them really well. But I didn't know this about her, that she really wanted to be a scientist during that late 19th century period, and just when natural history was really a big thing among normal people. So she made observations that lichens were a symbiosis between algae and fungi, and actually even came up with the idea of a symbiosis. So she went to the Royal Academy of Science to present this, and they booed her. And they would not allow her into this whole thing. And so she dropped out and wrote children's books. So, I mean, this is a great example of the injustices of science, yeah. But anyway, it's a good story to have, and I like to tell it so that to give her some credit for having one of the biggest finds probably in science and not getting credit for it. So I'm almost running out of time, I guess. So this is desert varnish, and it blackens some of these rocks. These are in some of the desert regions. And in fact, a number of natives have actually scratched petrographs into this varnish and had many images. And this is all due to some really precipitates of manganese and iron by really strange microorganisms. And so there are also microbes that were found in the Atacama Desert, which has very little precipitation. And the life found just generally goes into suspended animation for the very, few hours, maybe a year, where there's actually any kind of precipitation. I think this person is actually in suspended animation here, trying to find one. And then my favorite, which just is new, is that also in the Atacama Desert, there's these little rock caves, and there's spider webs. And the spider webs condense a little bit of dew, and the microbes are just loaded all over these webs, taking advantage of that dew water that just actually, well, this is a really interesting adaptation. It's the only one of its kind that I know of. Also, when it comes to extreme drying, the organism that is noted to be the most radiation-resistant form is Deinococcus. It's found in the soil. That's what it looks like. Deinococcus radiodurans can survive up to 10,000 grays of radiation. For comparison, 5 grays is lethal for humans. 2,000 grays will completely sterilize a culture of 10 to the 9th E. coli per mil. So the resistance to radiation apparently, is due to a really, I mentioned, unbelievably efficient DNA repair system. So that what radiation can do is break a double-stranded DNA apart. And so this bug here, it can take, its chromosome can be broken in 20 different pieces. And then when added back with nutrients, etc., it can reconstruct that DNA with high fidelity. Most organisms can't do that. They can't match the piece that broke with the right piece next to it. And so it doesn't have that kind of repair. However, we realize now that Deinococcus actually is resistant to radiation levels not found naturally on Earth. So it's generally believed that radiation resistant is actually from adaptation to desiccation or drying, which also breaks DNA. So we now think this is just a sophisticated organism that can repair its DNA after a drying episode. Extreme halophiles. I don't know how many of you have seen the Great Salt Lakes in San Francisco that turn red. The red coloration is due to a rhodopsin pigment like we have in our eye. It's also involved as a proton pump like I talked about. There's a group of square microbes. These are the only square microbes known. They're about a micron across that are halophilic. Again, there's a lot of salt flats, and again, this red coloration is due to a bacterial eruption. Generally, the kind of organisms grow in about 4 to 5 molar salt. They can't grow below 3 molar salt. It turns out that the same concentration of salt on the outside is in the inside of the cell, and actually the proteins are not stable in the absence of high salt. So this is a case where the proteins have adapted to high salt. So they're a unique organism. And again, the only square microbe that we know, it's Walsby square bugs, and C isolated it, and it's had a genome sequenced. It's a pretty interesting bug, so the shapes are there. And the big key in, if you're growing in high salt, is dehydration, how you actually deal with that, and you generally build water around the cell so as to keep it from completely drying. And the proteins, again, are adapted to high salt, and they use this kind of same light-driven proton pump to make ATP. So in summary of this now, why the range of habitat conditions for extremophiles are important in astrobiology is they may be analogous to environmental conditions. Obviously cold on icy moons, low nutrient oceans, sea ice and icy moons, dry environment, Mars, etc. High and low pH, I don't know, atmospheres, I mean, could there be microbes in the atmosphere of Venus, you know, where there's plenty of sulfur? I mean, there's a couple of my colleagues in astrobiology who think so. I'm not sure, I don't. Geothermal and hydrothermal systems, including those that I haven't talked about yet, so I'll talk about that later. And then early Earth analogs. Later on, I'm going to talk about how complex can a metazoan, that is a multicellular animal, get after long evolution in the absence of oxygen. And I'll show you some stuff. And I think that's it for this particular lecture. And so next time I'll kind of finish this up tomorrow morning, because I haven't covered a couple of little things like life in really low nutrients that I want to cover, and then I'll start origin of life tomorrow morning. So I just wanted to make sure I ended on time. John?
Yeah, so I had a question about the Wolf-Simon paper, which is the Arsenic Life paper. And you had several criticisms about it. Do any of these letters that have been sent in show that the fundamental claim of the authors is in fact incorrect, which is that the arsenic that's in the organism is in fact in the DNA, incorporated in the DNA, because that's really what they claim. And so has that been shown not to be true? It's evident from reading the paper that they themselves didn't demonstrate it. It was all which is the unfortunate thing about it. But in fact, has it been shown yet that it's not in the DNA?
If you read the BioEssays paper, that apparently experiment has been done before, and it's just, essentially the DNA and RNA falls apart. The bond is such that it doesn't really work. And so apparently that has been known. A lot of the criticisms that I have read, and if you go, there's tons of blog pages. I mean, it's unbelievable how much stuff. I think this is one of the first papers I know of in which the internet actually was the critic. There were literally hundreds and hundreds of responses to this paper. And a couple of them are very, very extensive. There's a biochemist at University of British Columbia who wrote an extensive review. And they criticized everything from the growth experiment to the kind of medium that they used, the fact that they didn't actually analyze their arsenic solution for the amount of phosphate. Somebody said the brand of arsenic that they used, that you bought, actually has this much phosphate in them, some X amount. There were just a lot of things like that that were not found. A lot of criticism about the organism. Why not try another organism to see, even from a different environment, a different species of the same organism? There's literally hundreds of, criticisms of this paper.
It's surprising given the fact that a couple of the authors are very senior astrobiologists, one of whom is the head of the ASU astrobiology node. But of course astrobiology will survive because astrobiology began at NASA based on two discoveries, one of which turned out to be demonstrably false in 1995.
Yeah, I think though that I think the ALH, the finding of Dave McKay, I think the interpretation was wrong, but I think their experiments were sound and their analysis was sound. I think the interpretation was wrong, and so I think there's a little bit of a difference there. Also, I think we have a very large young community in astrobiology now that are very upset over papers like that. And I can't tell you how many of our former students, current students, and students that I've interacted with at the astrobiology graduate student meetings that I've gone to that have contacted me about this. And in fact, I taught part of the winter school in Hawaii on astrobiology in January of this year. And that's all they wanted to talk about. I mean, really, let's talk about this stuff. And what does this mean for astrobiology? And is this going to set us back, et cetera? And so it was I had to spend a lot of time pep talking on this. But that's why I bring it up, because I think this is an example. If you read this paper critically, as a microbiologist, I would never have allowed that paper to be published. But I would have made some really strong suggestions on how to perhaps make it publishable. And I think that's one of the mistakes that was made. It was not reviewed well. There's only one microbiologist among all those authors, and that's Ron Ormland.
Yeah, but there are other astrobiologists who should know that.
Yeah, yeah.
Have a broad enough view that
Yeah.
it is very surprising. I don't think it's going to set back astrobiology, but it will make it harder to get papers like this accepted for review in science.
Yeah.
That's what the impact.
It's going to be a cold day in hell, though, before NASA comes up with a press release. We're going to have to find a really definitive exoplanet Earth for the next big, I think, NASA press release.
You mentioned that the present tree of life might be a branch of an extinct tree. This is a possibility you consider, or is there any indication? What are the
there's no evidence for that. I think the people that like it are the ones that don't believe that high temperature microbes were the early microbes. So I have some French colleagues, for example, who just don't believe that it can be a high temperature micro. And they use, as the one example they use that it might not be, I mentioned to you that high temperature microbes are capable of growing at high temperature because they positively supercoil their DNA. The enzyme that does that, which is called a reverse gyrase, gyrases twist things. And so a reverse gyrase is twisting it in the right side rather than a normal gyrase. Well, they looked at that protein from one organism and tried to relate where it was from. And they claim that its closest relative in terms of its structure is in an organism that doesn't grow above about 45 degrees. So they think the reverse gyrase actually came from a moderately growing organism. But why would a moderate growing organism actually make a reverse gyrase, as opposed to maybe some exotic gyrase that is closely related? I don't buy the argument at all. That's not an argument for eliminating a tree. There may be others. A couple of the other arguments is based on the rate of mutation that we see, the descent on the branches, where we get, let's say, maybe a 1% change in the ribosomal RNA that we make the trees up on approximately 50 million year timescales. So in order to get something really radically different, maybe we would be looking at perhaps, hundreds of millions of years to do that. Would there be enough time to actually do that? I think that's a bigger issue. Now, I mentioned this 50 million years. This is calculations based on only a couple of different kinds of organisms. It turns out that the archaea evolve, the ribosomal RNAs evolve much more slowly than bacteria do. So for example, we can see 2 archaea species that are totally different, have completely different metabolisms and everything, but have an identical 16S ribosomal RNA that we make the trees from. So all of the exceptions to the rule to the tree of life is really in the archaea, and that's where we really have a problem. And so we have to use other methods to actually classify these organisms at the species level.